For those of you who I haven't had the pleasure of meeting first, my name is Lale Welsh. I'm the Executive Director at NDF, and I would like to turn it over to Dr. Angela Leck. However, before I do that, I just want to boast a little bit about Dr. Leck. Dr. Leck has been with NDF for, this is, I believe, the 4th year. She is our scientific director and has been very instrumental in leading some out-of-the-box thinking with regards to how we should be moving towards gene therapy. And we're going to hear a little bit about that more today and in future weeks to come. Dr. Leck is an expert on muscle wasting diseases for over a decade. She did her postdoc at Harvard and is now at Yale University, working out of the Leck lab with her husband and partner, Dr. Monko Leck. She has several prestigious fellowships from the Australian government, by example. And so without further ado, I would like to turn this over to Dr. Leck and let her tell us about the work she's been doing with us and round up a summary of the work NDF's been doing, and then we can learn a little bit more about her.
Thank you, Lali, for that kind introduction. So without further ado, I'm really glad to be able to open up this annual speaker symposium series. So thank you for giving me this opportunity. Let me just see. So an overview of what to expect today. I'll be giving you a summary of where the field is at, as well as touch upon the projects and funding by the NDF and the areas that we think are going to be very important for the future direction of the field. So just as a quick refresher, so GNE myopathy, also known as HIBM or Nonaka myopathy, all the same disease. It arises when patients inherit 2 recessive mutations in the GNE gene, pictured here. The incidence is anywhere between 1 to 9 patients per 1 million, although more frequent in certain ethnic populations where there are found a mutation such as the Japanese one or the Middle Eastern mutation. It's more common in those populations. So the onset of muscle weakness and wasting, it occurs in early adulthood and has a relatively slow progression, beginning with the distal muscles and sparing of the quadriceps. And here is an MRI image you can see over time of a patient where the muscles are slowly degrading. There is also, when you look under the microscope, this characteristic rim vacuole appearance that seemed to be characteristic of GNE myopathy muscle. So, the GNE gene is expressed, can be found in your DNA. And as a brief refresher, that gets transcribed into RNA and then translated into protein, where the Genie protein can actually be found in many tissues of the human body, not just the muscle. Actually, when you look at the levels of Genie expression across all different tissues of the body, you can actually see that in the skeletal muscle, it's one of the lowest expressing tissue for the GNE gene. It's actually more highly expressed in the liver and the brain and the salivary gland. So it's actually really curious as to why the disease manifests in a skeletal muscle phenotype and doesn't seem to affect any other tissues. So indicating that the gene may play a very specialized role in the skeletal muscle. So the Genie gene encodes an enzyme with 2 functions, what's known as the epimerase domain and a kinase domain. And it's involved in catalyzing 2 different steps in the sialic acid biosynthesis pathway. So within the cell, there is a pathway that converts glucose taken up by the cell to sialic acid that then gets attached to surface proteins. And in this sort of chemical biological reaction, there are several steps, and the Genie gene, using its 2 domains, helps to convert UDP glutamate to MANAC and MANAC to MANAC 6-phosphate in these 2 particular steps. So the end result is sialic acid, and sialic acid will get activated into CMP sialic acid and then be attached to different proteins. So actually what happens is when you look at a muscle, the surface of a cell, different proteins stick out of it. And to these proteins, the cell attaches carbohydrate or sugar groups to these proteins that stick outside of the cell. And some of these involve sialic acid. And that is sort of where GNE, the big picture of GNE and what it does inside the cell. So the current thinking on why mutations in GNE results in a muscle wasting disease is that it reduces the overall enzyme activity, either the epimerase domain or the kinase domain, which in turn results in decreased sialic acid production, which causes a reduction in the attachment of sialic acid groups to muscle proteins in a process known as sialylation, which you may have heard of, and it's kind of a bit of a mouthful to say. So sialylation of muscle glycoproteins can actually be detected when you take a biopsy sample and you look at the cross-section of a muscle bundle, which, as you can see, is sort of like a bundle of spaghetti. And if you look at it face on, you can see using a lectin staining all the sialylation of muscle surface proteins by the green staining. And in muscle isolated from GNE myopathy subjects, you can see that there is a noticeably reduced level of the green staining around the individual muscle fibers. So this is what we think, at least the scientists believe, is the basis of the disease. So there have been several efforts to recapitulate the disease in mouse. The consensus in the field is that they each have their strengths and weaknesses, but that none of them are totally ideal. So there's been efforts to engineer the common Persian Jewish mutation by two groups, which unfortunately didn't result in a muscle phenotype, and neither did this common, the Japanese founder mutation. However, another line was established based on a different Japanese founder mutation, which initially did result in a muscle phenotype, meaning that it could recapitulate the human disease in the mouse. But unfortunately, for some reason, this was lost over time upon successive breeding and expansion of this line, and it couldn't be reproduced in other labs. So the two main therapeutic avenues currently being explored for GNE are enzyme supplementation and genetic therapies. So enzyme supplementation is a strategy that targets the sialic acid biosynthesis pathway, whereas genetic therapies try to tackle the root of the problem on a genetic level by either replacing the entire gene or correcting the mutation itself. So this is a summary table of the different clinical and preclinical trials in the field. Some have gone into humans, while others have only been tested on animals. And as you can see, there are the two broad categories: enzyme supplementation and gene therapy at various stages of development. So clinical trials with enzyme supplementation, which is taken orally, is a lot safer and cheaper than gene therapy approaches, which is why it's at a more advanced stage. So first, I will cover efforts regarding enzyme supplementation. So this mode of therapy basically assumes that the primary pathology or the disease itself is due to defects in the sialic acid biosynthesis pathway and reduced levels of muscle surface protein sialylation. So the idea here is to increase the overall sialylation levels by supplementation of the pathway substrates such as MANAC or sialic acid itself to kind of boost this whole biological reaction and increase the efficiency of this pathway. And there's been encouraging preclinical evidence showing that sialylation increasing therapies such as oral Manac or sialic acid, can in fact improve the sialylation and prevent or improve the progression of GNE in mice. And the main issue with this is, can they be taken up efficiently by muscle cells? And also, can they then be incorporated into the metabolic pathway, considering that it's something external we're introducing and it's not something that the cell made on its own? Can we trick the cells into sort of incorporating these metabolites or these substrates into the biological reaction? So using the Japanese mouse model that harbored the muscle phenotype when it was still sort of working, studies showed that oral administration of different sialic acid metabolites listed here to be a viable therapeutic strategy because it increased overall levels of sialic acid concentrations in both the serum and the muscle, as well as preventing atrophy, weakness, and improving muscle function. So this is the distance run, and this is a level of a muscle wasting biomarker, creatine kinase. You could see that the treated ones were lower. So this provided the preclinical results, basically, that form the basis of these clinical trial efforts using these various sialic acid supplementation strategies. So Ultragenyx, the company, developed an extended release formulation of sialic acid, and a phase 2 trial was conducted, and it resulted in what they thought was significant improvement of upper extremity strength in treated patients. But this unfortunately didn't translate into their phase 3 clinical trial, where it was shown to be safe, but conferred no functional benefit over the placebo group over a 48-week period. So despite they saw an increase in serum levels of sialic acid compared to the placebo, this didn't really result in much of a difference between the placebo and the treated in terms of the muscle strength measurements. So it's really difficult to tell why it didn't work. There could be a few reasons. Could it be that the trial was too short of a period, or maybe insufficient levels of sialic acid entered the muscle tissue? Is it also a possibility that the disease may be caused by more than a deficiency in sialylation? So it's really difficult to pinpoint why it failed. And so that's where it stands at the moment. So another enzyme supplementation effort is currently underway at the NIH, where instead of sialic acid, they're exploring Manac supplementation. So you could see sialic acid is down here in the pathway, and Manac is a little bit more upstream of that. So it's just another kind of substrate in the entire biosynthesis pathway. So Manac supplementation has been shown to restore sialic acid production in vitro and in vivo models. So a lot of this work was done at the NIH, where using the Persian Jewish mouse with the Persian Jewish mutation, Manac given to the pregnant mice increased the survival of the mutant pups compared to the untreated, and that also it manages to rescue the muscle hypersialylation in the Genie mutant mice. So you can see with this particular lectin stain, untreated, you can't see sialylation, and treated with Manac, you can. And so that was quite promising. So the next stage of the Manac trial is about to begin. This is information I took from the clinicaltrials.gov website. It says August, and it's a 3-year trial, randomized placebo-controlled. There'll be 51 eligible subjects receiving either Manac or the placebo 3 times a day, with follow-up visits every 6 months. And the primary outcome measures they'll be looking for will be changes in muscle strength measurements. And importantly, GNEM patients require a genetic diagnosis to enroll. So that is, you need confirmation of a biallelic pathogenic mutation in both copies of the GNE gene. So next is the next class of therapy I'll discuss is genetic therapy. So we are now, at an interesting time where we can fortunately explore correcting the root cause of genetic diseases. So genetic therapies for GNEM can include gene replacement therapy, which involves introducing a new copy of the gene, or it can involve editing the defective copy of the gene itself within the patients by introducing gene editing machinery such as CRISPR-Cas9. So as I mentioned, the process of gene therapy, to just to elaborate on what I said, it's more straightforward, where basically two defective copies of the gene can be compensated by introducing instructions to encode the healthy copy of the gene that can then function alongside the mutant ones. With gene editing, you are targeting the mutation itself by introducing repair components essentially to convert the mutant sequence to a wild type sequence. And the main differences between the two types of strategies are highlighted here in this table. Basically, gene editing approaches are mutation dependent. It's not a one-size-fits-all therapy, but it's feasible to design these for founder mutations that impact a lot of patients. So one changes your DNA, the other one doesn't. And both approaches have been tried in humans, but gene editing is a lot newer technology and so is slightly behind on regulatory approval. So regardless of which form of genetic therapy, they all require a delivery vehicle, either to deliver the healthy copy of the Genie gene or the repair components to fix the mutant copy. There are currently viral and non-viral based delivery vehicles explored in the clinic. Viral-based ones are made popular by AAV, adeno-associated viruses that have been used for SMA and Duchenne muscular dystrophy. So it's a one-time treatment that assumes patients don't have pre-existing neutralizing antibodies. And with the right serotype type of virus, it can be really efficient at entering the muscle, but it is very costly, particularly for adults, to use this type of therapy. Non-viral based delivery methods include things such as nanoparticles or lipid-based particles. And these are amenable to multiple treatments as they are not known to generate neutralizing antibodies. They're currently not as efficient as viral-based ones at delivering their payload. So AAVs are still a lot more popular, but a lot of development efforts are currently centered around improving non-viral based delivery methods so that they can be competitive with AAV. So to date, there have been two efforts to attempt delivery of a healthy copy of the Genie gene, one in a human patient and the other in a mouse model. So I'll be briefly touching upon both of these studies. So in the first one, there has been a liposome delivery. Liposome is kind of like a lipid particle, where inside those lipid particles, there are the instructions that encode the healthy copy of the Genie gene. And the second study was a systemic delivery of the Genie gene packaged in an adeno-associated virus and delivered into a mouse. So for this gene therapy in the one patient using lipid nanoparticles, the patient received 7 intravenous injections of the Genie gene, and where the gene was detectable almost 72 hours post-injection. There was a detectable amount of increase in different silylation, different lectin stains. Unfortunately, there was no consistent increase in muscle function post the 7 intravenous injections. It was sort of really largely variable, and there was no significant increase in muscle function reported after the treatment. So there, in terms of the systemic AAV delivery of the GNE gene. These studies were piloted by Stella Mitrani-Rosenbaum's lab, where she used the AAV8 vector with the human GNE gene. And she could still detect RNA expression of GNE 6 months later, which was a good sign, but she didn't measure protein at the time due to lack of an antibody. Unfortunately, there was kind of no phenotype to correct at this stage. So all she could do was sort of measure if GNE got in there, how much it was expressing and how long it was expressing for. And so the field of gene therapy is really converging on the use of AAV to deliver genes to correct rare diseases. So this is going to be the direction that a lot of NDF scientists are headed towards. So in terms of gene editing approaches, it's, as I mentioned, more suitable for founder mutations. So in our lab, we've explored the use of prime editing CRISPR technology to engineer the common Persian Jewish mutation into wild type cells. And you can see it's only about 11% efficient compared to an editing efficiency of a control gene, which we could achieve 70% editing efficiency. So efficiencies of editing can really vary depending on the gene and where the mutation is located. So this will need to be optimized. So gene therapy, some considerations going forward, regardless of whether it's a gene correction or gene replacement. We need to know what is the minimal efficacious dose for systemic expression of the healthy gene or correction of the endogenous gene. What are the potential adverse side effects of overexpressing GNE or receiving a large dose of the virus? What is the immune reaction, and what is the potential implications? Is it life-threatening? And the answer to that is yes. There's been some unfortunate adverse events in the field recently, leading to death. Also, do some patients have pre-existing neutralizing antibodies to AAV that will make basically the body reject the AAV and not be able to efficiently get into the muscle cells? And also there's the issue of manufacturing and treatment costs. To scale this up is logistically a challenge. Who's going to cover the treatment costs, considering it goes up to the order of millions? So these are just some practical considerations going forward. And I just want to say that it's not a problem specific to the Genie field. It's actually these are common issues for all gene therapy for other rare diseases. So currently, some bottlenecks we are faced with that prevents us from finding and delivering therapy in the clinic are as follows. First of all, lack of an animal model with a reproducible and reliable muscle phenotype. Also, the slow progressing nature of the muscle pathology in GNE is challenging in terms of designing a clinical trial and measuring whether a particular therapy has had a positive effect, considering that the disease moves so slowly, which is problematic. So, and because of that, we need what are known as good clinical biomarkers that we can measure in clinical trials to know if a particular therapy has increased a marker that's or decreased a marker that's related to the disease pathology, and to make the muscles look more like healthy muscles. So basically, this is something that the NDF has invested a lot in. And there's also the possibility that the GNE gene possesses function beyond sialic acid biosynthesis that we are currently unaware of. So that if we were to address its function in sialic acid biosynthesis by increasing that pathway, we're not really addressing the root cause of the disease. So these are just some sort of to give you insight into what scientists in the field are thinking. So due to these bottlenecks, the NDF has allocated funding and resources accordingly to develop more reliable animal models, have better disease biomarkers, formulation genetic therapies, establishment of a biobank, and formation of a patient registry. In terms of the NDF-funded patient registry, if you're a patient and you haven't yet registered, please do so. This registry is to catalog where people with this disease are located, and will give NDF and their clinical and industry partners an idea of how many patients there are globally. It's part of being clinical trial ready, to see where the patients are, how to contact them, what mutation they have, and what is the state and the progression of their disease. And this is just a map of where the patients are located in the United States, but we're collecting information on the global presence of GNE myopathy, and it's not limited to just the United States. So as I mentioned, a very important area of translational research is being able to measure gene function and its downstream effects. So this is going to be able to help us, help tell us if a therapy is working to fix the underlying cellular problem, and will also help us to further relate patient mutations to properties of Genie and sialic acid levels. So a German group is currently being funded by the NDF to develop methods involving radio labeling of a substrate of the Genie enzyme, which is this UDP glutamate, so that they can measure how much of it is directly being converted to MANAC, and thus reflecting the enzymatic function and efficiency of the Genie gene. Dr. Crow is currently screening different lectins that bind sialic acid on cell surface proteins to see which can be used as reliable biomarkers in response to therapies. And the LEC lab is currently developing assays to measure sialylation of a particular membrane protein known as NCAM as a readout of how efficiently sialic acid can be incorporated in this pathway to result in a functional outcome. So to expand more on the last assay, the NCAM assay I mentioned, we're actually using it as a tool to improve the interpretation of genetic variants or mutations in the GNE gene. So as you know, mutations in the Genie gene results in the disease. But what you may not know is that there are mutations that occur sporadically everywhere in the genome. So the challenge in the field is how can we tell if a change in DNA causes a disease or not, whether it's pathogenic or benign? And these sort of variants where we don't know whether it causes disease or not are known as variants of uncertain significance. And patients with DNA changes classified as VUSs, they can't get a definitive genetic diagnosis. So this is actually a snapshot of it. The pie chart represents all the mutations in Genie cataloged in a particular database known as ClinVar. You can see that the majority, more than 50%, are actually classified as variants of uncertain significance, meaning that the clinician doesn't know if it's causing a disease or not. So patients with VUSs will not be eligible to receive therapies, especially gene-targeted ones. So it's really important that we figure out what to do with these VUSs and how to better interpret them, how to relate mutations to the specific function of the GNE gene. And I hope I've conveyed that message across. Dr. Weisleiter at Ohio State University is looking at how mutations in Genie affects muscle membrane integrity and also their resealing capacity when injured. So he has a hypothesis that mutations in the Genie gene may actually result in changes to the membrane composition, which leads to compromised membrane repair. So here he tested multiple Genie patient lines from the Biobank and has shown a resealing, reduction in resealing capacity. And he's also looked at patient muscle biopsies from the Nishino lab and shown an increase in a particular stain that indicates compromised membrane integrity. So these are just more assay readouts that are going to be important as we head towards clinical trials with different therapies. So there is also two efforts NDF is funding to generate new Genie mouse models. So these are the Rosenbaum and Martin labs. Both are trying to develop a Genie conditional knockout mouse in the muscle. Stella's group, actually, Dr. Rosenbaum she is working on characterizing the Genie knockout in zebrafish as another model to understand the disease and test therapies. So there are also two labs working on different strategies to replace the Genie gene for gene replacement therapy. The Martin lab at Nationwide Children's Hospital is looking at co-delivering the Genie gene along with a muscle regeneration factor. And the Credell lab at University of Washington is looking at targeting expression of GNE gene in muscle and liver tissues. There is also an effort by the Japanese lab, Wakako Yoshioka, to identify existing compounds with the potential to increase Genie function. Let me briefly explain the rationale behind this project. So basically, in the sialic acid biosynthesis pathway, once sialic acid is made and is activated, it triggers what's known as a negative feedback loop, where it can bind to GNE, the protein, and prevents it from catalyzing more of the reaction. So it makes it inactive. This is known as a feedback loop. And Dr. Yoshioka has performed some computational simulation to find that this activated sialic acid binds a particular pocket of the Genie protein that changes its conformation from an active to an inactive form, which you could see here. And the idea here is that she wants to find existing drugs that can block activated sialic acid from changing the Genie conformation, so keeping it in its active state so that more sialic acid can be produced. And that's sort of the idea behind that. So the NDF also thinks it's important to establish a patient sample biobank, given that there is currently a lack of a reliable mouse model. And this is also to help with studies of biomarkers, disease mechanism, and testing novel therapies. And so this resource has been established at Yale and is openly available and shared with all researchers in the field upon request. So some of you may have kindly donated a skin biopsy at one of the patient conferences with Dr. Tassin Mozaffar or Perry Shea. So thank you for that. These samples were received at Yale, where the tissues were processed and cells grown out of that, and then sort of banked in liquid nitrogen, ready for experimentation. What we've also done is converted from the skin biopsy, we have isolated fibroblast cells, skin cells that we can propagate in culture. And we have converted these to, these are muscle cell precursors. So these are early stage muscle cells. And we've also managed to turn them into kind of a more mature muscle cell to do experimentation on. Using these samples, we have also performed various informatic analysis, such as whole genome sequencing, to characterize the entire genome of GNE myopathy patients, as well as their family members, as well as to begin asking questions about the genetic variability between patients, particularly the mild cases versus the severe cases in the same family. We've also performed RNA sequencing, so looking at changes in the transcriptome or the RNA that can then be used to build a Genie disease profile, and it's going to be useful as a clinical outcome measure. So we performed RNA sequencing on several of the samples that we received. This is just a preliminary run, but based on the data from this, we can see that affected, which is the green, samples cluster together, as well as samples from the healthy family controls. They cluster together also, so they're all right next to each other on this graph. And that means this is a really good sign, because it means that there are signals in the data that can distinguish between diseased and healthy samples. And this is really good. And when we drill down further, it seems to be that there are two strong distinguishing genes, one that goes up and the other one that goes down in Genie patient samples that can potentially be used as biomarker in clinical trials. Yep. So yeah, with that, I would like to thank the NDF team and the donors, as well as patients and their families for being involved in the community, as well as all the scientists that have been a real pleasure to work with, and of course LEC Lab members that work on Genie projects. Is there any questions?
Yes. Angela, thank you so much for that elegant presentation that sort of summarizes everything that we've sort of funded that's relevant to driving this solution forward. I know that there's a couple of scientists on the panel, among the panelists here today. Number one, Dr. Julie Crudell is among the attendees. And I wanted to introduce myself to you, Dr. Crudell, for some time now. So it's nice to meet you this way. And I know that you just also put a question into the chat, which I will read to you right now from the patients as well as the scientists from Dr. Crudell. Let me just say that if you do have a question, please do pop it in there, and I promise to read every single question. Let me see, how do I get this, Rachel? I'm not seeing how to look at the Q&A. Okay, from Dr. Crudell, here we go. For the MANAC clinical trials, were muscle biopsies done? She wants to know, was the sialation rescued? Is it possible the trials failed because the MANAC couldn't get into the muscles as well as in humans as it did in mice? That's always a problem, right?
The sialic acid. So the MANAC, yeah, it hasn't been done. I think she means the sialic acid trial with ultragenics.
Yes.
I don't believe muscle biopsies were taken as an endpoint in in phase 3. Apparently it was done for phase 2, but it was too variable, the results they were seeing. So then they just apparently didn't incorporate that into phase 3. And so they did measure levels of, free serum sialic acid, but yeah, that's an issue. Did the sialic acid, did it go in? And if it's in, could it be incorporated into the glycoproteins to silylate them? So that's, yeah, these are all good questions, and I kind of wish they did ask those questions, and hopefully in future clinical trials they will incorporate those. And I believe in the MANAC trials planned that they will be doing that.
Yeah, and in the interest of transparency and honesty, we are in discussions with Ultragenics to try to resurrect a prodrug that they've been talking about with us in with the understanding that they've actually shown much more promising results in canine studies. So Dr. Leck, myself, and a number of other scientists are in discussions with them so that we can, and as soon as we know more, we'll definitely have them maybe come on and do a presentation to talk about that. So Dr. Kutel, you'll be able to ask questions directly from them too. Another question that I have here is from a patient. I believe from a patient. It says, if Dr. Wakako was successful in finding a way to create this negative feedback loop, would this mean that muscle function could be regained because sialic acid is being produced again? Or would this only slow the progression?
I'm leaning more towards the latter. It would be similar, it would function similarly to if a sialic acid therapy were to work.
I see.
It would be increasing levels of sialic acid. And I think if muscles are already damaged, it's not going to reverse that. But who knows? There are a lot of things scientists don't know about this area. And we're continually finding new things every day, not only in the community of GNE researchers, but also in the broader community of muscle diseases. We're constantly learning. Does gene therapy, does it reverse the symptoms? Does it just prevent? So we'll see. We're learning as there's more clinical trials in the Duchenne space and things like that.
And I think that, to your point, that we do learn things every day. This is why there could be an interminable list of things that we could continue to learn. But at a certain point, Angela, as you know, we have to move forward with gene therapy, not so quickly as some people were pushing us, because we've learned from unfortunate mistakes of people or problems that people who went before us have learned, like you mentioned in your talk, the tragic instance at Audentes where 2 adult patients died. And I believe that was because of the toxicity levels. I wanted to ask about that myself, if you could elaborate for some of our patients, because that comes up often and we get calls about that. Gene therapy has been successful so far, to my understanding, in small areas like the eye or small organs, and in babies and small children. But in adults, where is the state of progression right now with gene therapy? Because the AAV is required, from my understanding, to go through the entire body, all the cells, can in fact have a negative effect. And I'd like Dr. Crudell to someday do a presentation on that too, because I know you're working on those AAVs. But Angela, what do you think about the state of that, and what steps are we taking to be safe?
So currently, the AAV, so there's 2 ways to deliver AAV gene therapy. There's systemic, meaning that it gets delivered to your bloodstream intravenously, and then it goes everywhere, it gets distributed everywhere. Or there is what's known as localized delivery, where you can just deliver it to the eye or to the liver. And that is considered to be safer because you require a lot less virus, basically. And so the way that the dose, how much virus is determined, how much is needed for a particular patient, is based on their weight, how much virus per kilogram of the patient. And obviously, the heavier you are, so if you're treating an adult population, you're going to be getting larger volumes of this virus, greater absolute amounts of this virus. And we don't yet know if there is like an absolute ceiling as to, okay, this is the maximum you can be dosed with in a human before the liver just malfunctions. Because the thing is, even though we're using viral vectors that are targeted to the muscle, the majority actually goes to the liver. And if you and the more like about 90% or even more goes to the liver. It's just where it ends up, where things end up. And I think if you have a bad liver, that's going to be problematic. And the issue with those patients that unfortunately passed away from the Audentes trials was that they had underlying liver issues, and their livers just couldn't handle the gene therapy. And so
you're saying it was compounded by the fact that they had a comorbidity or something,
yeah, that's true.
pre-existing.
Yeah,
so that's not going to always be the case.
Yeah.
Yeah. Okay. And yes, that is an important point.
To my knowledge, there hasn't been a systemic delivery of gene therapy in an adult. And this is going to be interesting to see how much AAV we can pump a full-grown adult before we start seeing absolute, toxicity in the liver and things like that.
So can you talk about the steps that the scientists at NDF, are taking in order to address that issue?
Mm-hmm. Well, we are planning a large-scale toxicity study with a CRO, a company that can look at overexpressing the GNE gene at different doses, at escalating doses, and then measuring, harvesting different organs and measuring different sort of immune markers to see what kind of, toxicity, whether it's acceptable. And then we're going to run it by the FDA to ask them, is this okay? And before we have plans to dose a human.
Great. So I just wanted patients to hear that we are taking those steps. And to that point, and there's lots more questions, and I'll turn her over to that. But to that point, if you could sort of wrap up the overarching, the process that we're taking, the speed with which we are going towards GNE gene therapy. Could you please talk about the steps involved and how, and then I promise the patients that we'll have a separate presentation about that. But in brief, say where this fits into the journey towards gene therapy.
Well, the NDF are funding labs, academic labs, to work on different Genie gene therapy, but they are sort of considered, next generation gene therapy vectors. As I mentioned, PI Crudell is doing like a dual promoter. PI Martin is doing the dual gene, and these are sort of not very not very straightforward. The most straightforward approach is, just a single promoter and a single gene. And that's kind of what's been approved by the FDA. And in order to get to the clinic very quickly and for the fastest approval, the NDF wants to push for just a single promoter and a single gene, in the process of partnering with different companies to lead this effort, along with the regulatory experts to help us guide through the process of creating an IND and meeting with the FDA to see what's acceptable and what's not in a small pilot study. But I believe Rich is going to talk more about that next week.
Rich Corrigan is going to demystify that process for us next week, June 11th. So please do tune in for that. Thank you for that. And of course, Dr. Crudell, you'll be given an opportunity to explain to you what problem you're solving with your work at your wonderful lab. I do have a question from a patient, one comment and one question. One is that she wanted to let everybody know that she felt that the sialic acid extended release did in fact help her in spite of the failure to meet its endpoints in phase 3. And I hear this quite often from patients that while they were taking it, they really did show improvement. So again, this is a conversation that's not dead. We are resurrecting this conversation, and science is tough like this. Sometimes it doesn't produce the result that you need, but that doesn't mean to say that we're not going to continue to keep attacking from a different front.
Yeah. The issue is, how do we measure that? The companies need, and the clinicians need, a way to be able to measure the improvement. We need a number. We need to, because otherwise, we need statistical sort of significance associated with these various measurements to make sure that it's real and reproducible. And the companies aren't just sort of going ahead and producing a drug that may or may not work or works as like a placebo because patients really want it to work. So then it works in their head.
Yeah.
I'm not saying that's what's happening, but it needs to be measurable.
Isn't it something like 30% of the time placebos work? So,
Yeah.
you know, whatever works, works. But anyway, her next comment is that she has a question saying, in what way does gene editing change our DNA? And that's a question, again, that we've heard before. So if you could maybe shed some light on that.
So the way gene editing works is we want to target the editing machinery to the region of your gene that has the mutation. So, and then change that mutation to a healthy DNA, to a healthy DNA sequence. So it's targeted, it's really specific. We're directing it to exactly where your mutation is that's causing your disease, and we're changing the DNA to make it a healthy copy. Does that make sense?
It does to me.
Yeah.
The patients can weigh in if they have follow-up questions on that for that explanation. I have a question from Mangesh, who's a patient in India. Hi, Mangesh. He says, I understand that the AstraZeneca and Johnson & Johnson COVID vaccines use adenoviral vectors for delivery. Does this have any implications for AAV-based gene therapies in terms of pre-existing neutralizing antibodies?
So they're using adenovirus vectors. That's different to AAV. AAV is adeno-associated virus, and the two are completely different. They're really, really different. So there is no, as my understanding goes, there is no implication for developing existing neutralizing antibodies.
So the antibody will not affect anything related to gene therapy for GNE.
No. It's easy to confuse the two. It has that same word, adenovirus, but it's different. One is they're very, very different. So that's my answer.
His follow-up question is, have the two different gene replacement therapies, liposome versus systemic via AAV, been tried on other muscle, I think you answered this, on other muscular or not diseases, and have any differences in efficacy been observed? Wondering if AAV gene therapies have been shown to be more effective compared to liposome delivery.
Yeah, it has. It definitely has been attempted. And currently everyone is kind of converging on AAV gene therapy because it's shown such great success in SMA and in DMD. Those are two other muscle diseases. It's liposome is not as effective for systemic delivery, especially muscle targeting. So yeah, they have been tried.
Okay, good. And then Dr. Cradell's correcting something. She says 3 pediatric patients died in the Audentes trial, no adults enrolled in Audentes trials. So thank you for that.
No.
Another question from another patient. Have there been any studies on whether myogenic cell transplantation on stem cell therapy would be effective in GNE animal models or in patients? She's a first-year medical student. She wants you.
I don't think anyone has or is pursuing that right now in the GNE field. I know, this is something that's been attempted for many different muscular dystrophies, but has not yielded success. I mean, it is the holy grail, actually, because that allows you to generate new muscle. And I believe it will be achieved sometime in the future, but we are not there yet. And no one currently is working on it for Genie, but I believe that if someone were to find a way to efficiently transplant new muscle cells, we could do it for Genie patients. Like, it could be translated pretty rapidly from like the Duchenne field or something else into the Genie field. The strategy would be pretty translatable.
And it's pretty exciting that just even to think about the possibility of that now. We obviously rely on our coterie of 40 some odd scientists to let us know when they think that there's some merit to pursuing that. Just so you guys know, that's how NDF operates. We do not claim to be scientists. We claim to be a platform to expedite and to collaborate among scientists so that they can build on top of their own understanding that they build from one another. That's something that NDF is absolutely committed to doing. Dr. Kudelka continues, and I apologize for jumping around because I'm seeing these as they come in. There have been systemic deliveries in adults with liver disease or hemophilias. It's just that their doses are 10 to 100 times lower. That's what Dr. Kudelka, have any comment? Not yet. Thank you for that, Dr. Kudel. And then other patients are commenting and thanking you for a lovely presentation. There's tons of those kinds of comments. Angela, do you have any closing remarks or anything that you want to wrap up and say?
No, just to keep stay tuned and keep being involved in the community, and because we're all working together here, and everyone has a role to play. As you know, my husband also has a muscle wasting disease, and we were at some stage patients just like you guys. We didn't know anything about muscle diseases. We went into this completely blinded, and mind you, very naive, thinking we could fix the thing. But the point is, don't just sit back and wait for something to happen. Everyone has a role to play, and everyone can help in some manner. So please, whether it's helping to fundraise, whether it's donating samples or outpatient outreach, please stay involved with the NDF community, and keep in touch with your fellow researchers and other patients, and in the community. So thank you.
Thank you for that reminder. And just so you guys know, that the scientific funding of research is just one program at the NDF. It's a very, very key and important program, but we also have many other programs, patient-facing programs. While we are waiting for some of these results to come in, we find it absolutely our responsibility to as part of our mission, to make the lives of those people who are living with it or in fear of getting it a lot easier. And we provide practical day-to-day solutions in a myriad of ways. And I encourage you all to go to curegnem.org. This is our new website. Go straight there and take a look at our programs and see which ones benefit you. And we're always open to hearing from you about how we can add to our programs and list of things. We are here just for you. We are very proud of the collaborations that we have forged among the scientists, and we're doing this on behalf of patients around the world. Final question is about where people can go for adding their information to a registry that just came in to my purview. Angela, I just want to say before you answer that question that there are a number of registries out there, and we are all in the process of merging those and sharing that data on behalf of the patients, because that's what's in everyone's best interest. In the meanwhile, Angela, where do you recommend people go? To the
there is, you go to the QHIBM, now it's GNE. There is a link to the registry. And I think there is an email too. So sorry, I should have put in those details, but it's definitely on the website and you'll find it.
How about we get you guys that information moving forward, because no matter where you put that information, we are going to be collaborating with other nonprofit groups to make sure that we share that data. But in the meanwhile, Tara just told me it's registry at curegnem.org. If you have any questions, concerns, comments, we're always here for you. This is our only reason for existing. So again, thank you everyone for participating in this first kickoff meeting. Really appreciate, Angela, your presentation. And I look forward to seeing you all next week when we hear from Rich Horgan, who is going to talk more about the gene therapy process and the business development piece of it, which I think is incredibly interesting and you guys should know about. So again, thank you for everybody's participation, and I look forward to speaking to you all next week. This meeting has been recorded, and a copy of this will be made available at curegnem.org. Thank you, everyone.